Good morning, grade 11s. So this is lesson 5 of the urinary system. I didn't get any questions from anybody yesterday, so I am presuming that lesson 4 went okay and that you understand lesson 4. So today what I'm going to be talking about is, yesterday I gave you an overview of how urine is formed. And today I'm going to go back and I'm going to give you the formation of urine in a little bit more detail. So that's what we're going to do today. And you need to send me questions or answer questions if there is stuff that you don't understand. Okay, so this particular drawing looks at the interface, in other words, the place where the two parts lie together, between the wall of the capillary of the glomerulus and the lining of Bowman's capsule. So if you look carefully here, this is the wall of the capillary. So here's the capillary lumen. In other words, this is the blood within the capillary. This, what they've shown in blue here, is the two layers of basement membrane. Remember, one of them comes from this squamous epithelium and the other comes from this squamous epithelium. Then these are the little foot processes, as they call, the little extensions of the podocytes. So remember that I said to you that podocytes are shaped a bit like starfishes. So if you take a section through them, this is what you're going to see. You're going to see basement membrane, and you're going to see these little things. And then between them are slits. So here are little micropores, and here are little slits. Okay. So the basement membrane, here it's called basal lamina. You just need to know it as basement membrane. Two layers of basement membrane. This is what acts as the filter. The micropores and the slits are actually quite big, and because they're so big, they don't act as a filter at all. What does act as a filter is the little fibers in the basement membranes. So let's have a look at the text. The text just shows you exactly what I've said to you. There are micropores in the squamous epithelium of the capillaries, and there's slits between the podocytes. So that the only thing that really separates this blood liquid, I'm going back to the drawing, from the liquid of the filtrate on this side here, is really the two layers of basement membrane, because this is quite big and this slit here is also quite big. So the two layers of basement membrane act as a filter. They only allow very small molecules to filter from the capillaries into the lumen of Bowman's capsule. And because the molecules that pass through are very small, the process is called ultrafiltration, because they are ultra small. So ultrafiltration occurs in the Malpighian body. And remember, the bore of the afferent arteriole is bigger than the bore of the efferent arteriole, so more blood can enter the glomerular capillaries and can leave. Doesn't matter how many times I read these things, it looks correct, and then I see the errors when I'm actually going through it with you. I apologize. So the large number of capillaries serves to increase the surface area for maximum filtration. The fine meshwork of the basement membranes prevents any large molecules such as proteins and obviously any cells like blood corpuscles, etc., from leaving the blood in the capillaries and passing through. The liquid that passes into Bowman's capsule is called the filtrate. And the filtrate at that point contains water and many solute molecules such as salts, glucose, urea, and amino acids. Blood platelets, blood corpuscles, red and white blood cells in other words, and blood proteins, which are large molecules, together with some of the water and some of the urea and waste substances, remain in the blood vessels of the glomerulus. Okay. Then the filtrate is now in the proximal convoluted tubule. Remember, the tubular reabsorption is the passing of useful molecules, which were small and filtered and have the potential to obviously pass out in the urine, the useful molecules need to be reabsorbed. So as the filtrate passes along the nephron, all the useful molecules such as glucose, amino acids, vitamins, minerals, and most of the water are reabsorbed back into the blood. It occurs mainly in the proximal convoluted tubule, which is structurally suited for this process. So the proximal convoluted tubule is the longest and widest part of the nephron. Its walls are, so in terms of length, and remember, stuff that is in blue, you don't have to learn. So 14 millimeters long and 60 micrometers in diameter. Obviously typed an O instead of a 0 there. Sorry guys, that's meant to be 60. Its walls are formed of a single layer of epithelial cells with many microvilli. Remember, look at the micrograph, many microvilli, that also increase the surface area relative to the volume of the lumen. And because there are numerous mitochondria, they provide the energy for what we're going to talk about, active absorption. So a lot of the absorption, so this is the lumen here, and this is the filtrate. And remember that the filtrate contains lots of different substances, but there's still some small useful molecules in there. And they are going to pass from the filtrate through this layer of cuboidal epithelium and into the surrounding capillaries. And that is tubular reabsorption. They're going to pass back into the bloodstream. This is a scanning electron micrograph showing the proximal convoluted tubule. And what you can see is the many microvilli that are present there. It's in the kidney of a rat, and the magnification is 3,200 times. Okay, just for interest, because it was beautiful. So, very simplistically, and I think that your textbooks got quite a lot of detail in your information. We are not expecting you to know that degree of detail. So the simple version, which is what I've done on this PowerPoint, is the version we need you to know. So remember that glucose molecules were small, so they passed out of the glomerulus with glomerulofiltration or ultrafiltration. And glucose is useful to the body. It's a source of energy. And so in a person with a normal balanced diet, a normal healthy person, non-diabetic, all of the glucose is reabsorbed, as are all of the amino acids and some of the minerals, a lot of the minerals, or the salts. Some of the reabsorption is passive. I think back to what you learned about last year, diffusion and osmosis. You learned that diffusion and osmosis are passive processes. In other words, the molecules simply move from a region of higher concentration to a region of lower concentration. And it doesn't require an energy input. However, some of the reabsorption of certain of the molecules is active, and it requires the energy supplied by those many mitochondria in the proximal convoluted tubule cells. Now, you don't have to know what goes passively and what goes actively. You don't need to know that amount of detail. And so just remember, some of it is passive by diffusion and osmosis. Some of it is active and requires the energy from the many mitochondria. And that's the degree of detail I expect you to know. And then the filtrate continues. Tubular secretion, which remember is the process whereby certain of the molecules that are toxic remained behind in the blood at the glomerulus because they were large molecules, but they need to be excreted in the urine. So tubular excretion is when those molecules are passed from the blood in the peritubular capillary network into the tubule and then become part of the urine and get passed out as urine. So that is called tubular secretion. And it occurs mostly in the proximal convoluted tubule. So substances that didn't filter from the blood in the glomerulus, but should be part of urine, are moved from the peritubular capillaries into the lumen of the tubule and join the filtrate. So tubular secretion usually removes substances that are too large to be filtered. For example, certain antibiotics and toxins, or those that are in excess in the blood. So hydrogen ions and potassium ions. The loop of Henle. Very simplistically, and please don't go and look this up anywhere because it's quite complicated in some of the sources that you'll look at. So its main function is the reabsorption of water from the filtrate. So the filtrate that passes through from the glomerulus into the tubule contains an extremely high volume of water, and most of that water is reabsorbed. And I'll give you some figures later on so that you can compare the volume of filtrate versus the volume of urine that is produced in 1 minute. And so a lot of the reabsorption of water occurs, in fact, in the loop of Henle. Sodium chloride in the forms of sodium ions and chloride ions also pass out from the filtrate in the loop of Henle. And that's important because it helps to conserve water for the organism and it makes the urine more concentrated. So loop of Henle, reabsorb water from the filtrate, and sodium and chloride ions pass out of the filtrate and go into the fluid of the medulla. Okay, there's no regulation of how much water passes out of the filtrate in the loop of Henle. So the body has no control over it. It's just water is reabsorbed. But there's no regulation of that. That's going to happen in the distal convoluted tubule and in the collecting duct. All right, so homework. Complete activity 3, page 245 of the textbook. In addition, complete activities 5 and 6 on pages 251 and 252 of the textbook. Okay, I'm going to leave it at that today. And next lesson, I will start with osmoregulation. So do those activities and we'll carry on next time. Okay, email me or Ms. Florenza if you've got any problems, any queries. Okay.